In the first video in Module 4.1, I gave you an overview of the cell cycle and started focusing in on mitosis or M phase. In the second video, we looked at some of the details of M phase, and now we're going to head back out and we're going to pull out again and basically look at the bigger picture of the cell cycle, and in particular about these things called cell cycle checkpoints. And I'll talk about 3 checkpoints. There may be more depending on how you count. We'll talk about 3 of them, and they're going to control the progression through the cell cycle. And we'll talk about why it's important that we control that progression as well. All right, so let's reorient. This is a slightly different picture, but it's still just showing the cell cycle. So you can see interphase is most of it, and that's everything from G1 to S to G2. M phase includes both mitosis and cytokinesis. And what is being added here is basically, we're trying to make this look a little bit like a thermostat or just a wheel that you can spin, but there are certain blocks on it that have to be removed before you can actually continue. And so we have something that is called the G2 checkpoint down here at the bottom. And this is something that stops the cell from entering M phase. And so until that G2 checkpoint is removed, the cell can't actually enter mitosis. The M checkpoint happens at the end of mitosis, and it prevents the cell from actually dividing or leaving M phase. And so there you go. And G1 is just something that is present before the cell can actually enter the DNA synthesis phase. So we've got these 3 checkpoints. I can tell you a little bit about what happens at each. So at G1, the basic idea is the cell is asking, is the DNA damaged? So that's going to happen before the DNA actually gets replicated, which sounds like a useful thing. The G2 checkpoint is sort of a resource check. So it's now asking, is there enough food available for mitosis? And if the answer is yes, that removes this checkpoint and the cell can now enter M phase. And then the M checkpoint happens actually sometime during, say, like metaphase or thereabouts. And the basic question is, are the centromeres actually connected to the mitotic spindles? And if they are, then you can start pulling them apart and creating 2 new cells. One nice website for this, you can go to this Howard Hughes Institute website, HHMI, and they actually have a really cool overview of these different checkpoints. You'll see they actually include 4 checkpoints in here. Your book only really talks about 3 of them, but still, it's a pretty cool resource that explains how the checkpoints work and what they do. Okay, so now that you've seen the checkpoints, let's talk about a couple of other mechanisms that are used to control the cell cycle in most cells. So one of these things is called anchorage dependence. And what this basically means is just that most cells can only divide when they're actually attached to a surface. Now, sometimes this surface is other cells, sometimes it's other materials, like here it's attached to the surface of a petri dish. But this anchorage dependence means that a normal cell will not divide unless it is attached to something. Another one is contact inhibition. And so in this case, a normal cell, when it is at a certain density, so basically when enough of its membrane is touching the membranes of other cells, it will stop dividing. So contact inhibition is what it sounds like. When the cell comes in contact with enough other cells, it will stop dividing. And then there are some others. One kind of cool experiment here, there are some others, but one cool experiment that is often used to think about contact inhibition in particular is this thing called the scratch test. So with the scratch test, you're basically going to take this knife and you're going to scrape it across the surface of this petri dish. And that's going to damage the cells that are at the center where you've scraped. And it's going to provide some room and reduce the contact inhibition. And so what happens in a normal cell is that, or a normal set of cells, is that they will divide until they are once again all butt up against each other. And so once that happens, they should stop dividing if they're normal cells. The big problem is what happens when cells stop paying attention to those rules. So what happens when the cells no longer need to be attached to a surface in order to divide? That's already a bit of a problem. And then what happens if they start ignoring their neighbors? And so here I've shown that as basically all these cells are now piled up on top of each other. And in fact, they could just be floating around in the fluid and continuing to divide. So I'll try and address what happens in the next video, but maybe think about that a little on your own first.